Hey, what's up? So today we're going to talk about a study published in the New England Journal of Medicine that's actually pretty recent about a new technique in medicine that's been used pretty much by the time to cure HIV on time. So we have some information about gene editing of CCR5 in autologous CD4 T cells of persons infected with HIV. So in the background of this study we have some very interesting information. So CCR5 is the major co-receptor for human immunodeficiency virus, so which is called HIV. So these people investigated whether site-specific modification of the gene. So this is why the method it's called gene editing. The infusion of autologous CD4 T cells in which the CCR5 gene was rendered permanently dysfunctional by a zinc finger nuclease. So it's kind of a way to say that it's a pretty much safe method because it has a very solid base scientifically speaking. And I got a video for you that's about a news. This was very recent actually. That's a case of a boy that was actually cured of HIV thanks to the gene editing. So
This is a technological tour de force out of the University of Pennsylvania. And it's based on the fact that an HIV virus enters a white cell and infects it, a T cell, by a protein that's on the surface of that cell, okay? Without that protein, it cannot get into the cell. It's locked out. And what these researchers did...
So I'm going to make a stop right there because this is an actually important step on the gene editing. So what they are looking for actually in this with this method, it's to stop or to avoid that the virus can get to the cell. How they do that? Destroying or submitting some receptors that are specifically made for this kind of virus. So let's see a little bit more what the doctor is explaining right now about this process.
Was they took the white cells from somebody with HIV, they brought them outside the body, and then imagine this, out of 20,000 genes that are in a cell, they found the one that controls for that protein on the cell surface. They disabled it, they put them back into the person, and now the person had the HIV virus circulating but couldn't get into the cells to infect it.
I think this is fascinating, right? Because this is the future of medicine, is gene therapy and genetics. And in this specific case with HIV, it was this mutation called CCR5, that if you inherited that from both parents, it's very rare, you wouldn't get HIV. So scientists found that and said, let's make this happen. How successful is it? Does it cure you of HIV?
Well, they're not using the word cure, but this is a small safety study, okay? And there was 12 patients, 6 of them they could really look at because they could stop the medication, see what happened. In 4 of those, the HIV levels went down. 1 of those 4, it actually was undetectable.
All right, thank you.
This is really, I agree with her, extraordinary stuff. And in fact, there was only one gene, which makes it much easier.
Astounding.
Thank you, John.
So many important things are being said in that interview with a doctor that was actually proving this method. But there's something really important that he said about it's not actually called a cure because it can be applied to everybody. So it would be a kind of a concept mistake for some people. So we have to be really clear on that. So it's actually not a cure, it's kind of more than uh, a very personal concept. It's actually for me uh, a very efficient method to avoid the infection or avoid infection progress on time. So how returning to the study, so these people enrolled 12 patients in an open label, non-randomized, uncontrolled study of a single dose of the ZFN modified autologous CD4 T cells. So the patients had chronic viremic HIV infection while they were receiving highly active antiretroviral therapy. That's actually the kind of therapy or medicine that's been taken by all these people that's infected by the HIV. So gene editing it's trying to replace this. We're all hoping for the best to they accomplish that. So six of them, that mean that 50%, underwent an interruption in antiretroviral treatment 4 weeks after the infusion of 10 billion autologous CD4 T cells. 11 to 20% of which were genetically modified by the ZVN. So you might be wondering why this ZVN is actually being named at every time. It's because, like I said before, this is kind of the base of this whole method that goes by that goes really related to the discovering of the CCR5 gene. So that's kind of the importance of this. The primary outcome was safety as assessed by treatment related adverse events. Secondary outcomes included measures of immune reconstitution and HIV resistance. So it's they are trying to say it's that it's not actually changed at all at that time. So we have here I got you another video to explain kind of the virus progress. So we're going to take a look at this. It's kind of long, but we're going to try it.
CRISPR is a new nuclease-based approach to gene editing that makes use of a bacterially derived protein to introduce a break at a specific location within the genome. A plasmid is introduced into the cell, where upon entry to the nucleus, the components necessary to perform CRISPR gene editing are manufactured. The two components of
I'm going to pause it right there
CRISPR are the Cas9 protein,
because there's something really important about it. That's what the doctor in the interview was explaining as well, that this method is actually not killing the virus, it's just avoiding that it gets to the cell. And in the worst of the scenarios, if it gets to the cell, it can get in to the cell. So that's kind of two concepts or two ideas that we have to be really real clearly. So I'm going to continue the video, so I hope it helps.
which cuts the DNA, and a guide RNA, which directs Cas9 to a specific location where the cut is to be made through hybridization with its matching genomic sequence. While bacterial Cas9 is capable of introducing double-strand breaks, a modified version introduces only single-strand nicks to the DNA, which can be exploited to reduce the off-target cuts sometimes seen using the wild-type Cas9. By using the nickase form of Cas9, with two guide RNAs targeting opposite strands of the target DNA, a double-strand break is achieved with reduced off-target activity. Horizon is offering CRISPR as a standalone system and also in combination with RAAV in order to achieve gene editing efficiencies not possible with either technology alone.
So before we go to the results, I have to make um, a point of order. In fact, that video was taken from a company that's actually dedicated to the gene editing, but it's not my intention to make some publicity. But I thought that it would be really helpful for you, all of you, to understand how actually this kind of method works, and in which part of the process of the virus progress it actually acts. So the results of this study were actually very positive, saying in a way. So one serious adverse event was associated with the infusion of the ZVN modified. This is actually really important because the term ZFN, uh, it actually doesn't occur to many for us, but when we're talking about a ZFN modified autologous CD4 T cells, we're talking about what's the method changed. So I was telling you, and it was attributed to a transfusion reaction. So the median CD4 T cell count was of 1517 per cubic millimeter at week number 1, a significant increase from the pre-infusion count of 448 per cubic millimeter. So the median concentration of the CCR5, this is what it really matters to us, because this is what we're really trying to understand. The median concentration of the CCR5 modified CD4 T cells at 1 week was 250 cells per cubic millimeter. This constituted 8.8% of circulating peripheral blood mononuclear cells. These all mononuclear cells are the part of our immunity system, so we have to be clear in that. And 13.9% of circulating CD4 T cells, modified cells, had an estimated mean half-life of 48 weeks. So resuming all this, we could say that results have been really positive, and we're all hoping for the best. So this kind of method would be available, I believe, soon. So thank you all for your attention. I hope this video would be really helpful, and we'll see you in the next occasion. Bye.